Let's look at number 3. This is a good answer for histidine. This is a good answer for glycine. And this is a good answer for proline. Good. Those are all important ideas. Let's look at number 4. Okay, so what do you call the bonds between amino acids and a peptide? It's called a peptide bond. Very good. But look at number 4. The next part of my question was, what functional group does the bond consist of? Now, you said it was a carboxy group plus a nitrogen. Now, first of all, that's not really quite right. We probably shouldn't call, so here's the peptide bond right here. We shouldn't call this a carboxy anymore. Sometimes I call it the carboxy carbon, but it's not a carboxy group anymore. You could call it a carbonyl group, a carbonyl and a nitrogen. But in any case, this isn't what I was going for here. Do you remember when we talked about this? There's a specific name for this type of functional group in organic chemistry. What's the...
Amide.
Say again?
Amide.
That's what I was going for number 4. All right, I think that's something you could be tested on. So this is called an amide. What's the name for this functional group at the end of the chain?
Amine.
Amine. So we don't want to confuse the amide and the amine. Okay. Number 5. So that's a good answer for A and B. Now remember that for B, histidine is only 10% charged. Histidine is only 10% charged, but that's still enough for it to be put in the positively charged category in your textbook. Okay. Is there any particular reason why you said proline? So I'm looking now at part C. Is there any particular reason why you said that proline was aromatic?
Because it has a ring structure in it.
Okay.
But then again, that's not the side chain. It's part of the main chain.
Yeah. Well, it's tricky. The ring is part of the side chain, but it intersects with the main chain. The ring is part of the side chain, but it intersects with the main chain. All right. So are you saying that all rings are aromatic?
No.
Do you remember that we went over aromaticity in your organic chemistry class? We did about 1 or 2 sessions on that. Remember Huckel's rule? We talked about how you had to count the pi electrons. They had to be like in a list of like 2, 6, or 10 electrons. Does that ring any bells?
Yes.
Okay. Well, we don't need to go over all that material from scratch right now. But the point is that definitely shows us not all rings are aromatic. Do you remember one thing you have to be aromatic is a bunch of double bonds? I'm not going to go over the details. So is proline aromatic?
No.
How can you tell it's definitely not aromatic?
Because it doesn't have any double bonds.
At all. Okay.
Yeah.
All right. So should proline be in this list for part C?
No.
Okay, that's right. Good. Proline should not go in this list. It's definitely not aromatic. If I was asking you for everything that has a ring, maybe proline should be in there, but it's not aromatic. This is an important question. Again, this is a pretty typical type of test problem, so this is an important one to digest. All right, proline should not go here. Let's see, your answers for parts A and B were correct. I'm going to take a look now, number 6. So this is the type of problem that might be better to do on your drawing program so that you can fit the whole thing in. Or the other thing I would recommend is when you're doing this type of problem, I recommend turn your page lengthwise like this, because if you turn it this way, you can fit a longer chain in, if you see what I'm saying.
Yeah.
Okay. Any case, let me see what you have here.
I got the hard way.
All right, that's a good picture for tryptophan. I have a hard time remembering how to draw tryptophan, but you got that correct, and it is aromatic. So these are good pictures for glutamic acid and glutamine. I often get the wrong number of carbons and confuse them with aspartic acid and asparagine, but you did those correctly. Okay, did you do this whole thing from memory?
Yes.
That's good. There's a good chance you might have to do this on the exam. A common type of exam problem is just like this. They'll write out like a 5 or 6 or 7 or 8 membered, or maybe just 3 or 4 membered peptide and ask you to draw the whole thing out. So that's an important thing to make sure you know how to do. I asked you to get the charges right too. Let's see. Okay. So your example here for proline is incorrect. Excuse me. When we practiced this in our last tutoring session, you got this right, but I think in this picture we got it incorrect. Take a look at what you have written here. Can you see what went wrong with your picture of proline?
Oh, I see. The NH3 plus is not supposed to be there. It's supposed to be there's supposed to be a link from, or a bond from the NH group down.
Yeah, so the key thing is, how many nitrogens are there in the entire proline residue?
1.
Not 2. That's the whole thing that's special about proline. The whole thing is not that, it's not just that it has a ring. Lots of amino acids have rings. The thing that's weird about proline is that the ring includes the main chain. Excuse me. Thanks. The thing that's weird about proline is that the ring includes the main chain. All right, so all right, let's practice that. Try to do this again without looking at your notes from memory. Try drawing this tripeptide on your screen.
Okay.
And we'll draw it at physiological pH as usual.
Okay.
Okay. Point to the nitrogen in the proline. Yes. How many hydrogens is that attached to?
Should be attached to just none.
Why?
Because now it's neutral.
And how many bonds does a neutral nitrogen form?
3.
3. Point to the nitrogen in the ASN, the main chain nitrogen. Why does that nitrogen have a hydrogen and the proline nitrogen doesn't, if they're both neutral?
Because this one has 3 bonds already. And so does the proline.
Okay, well, you didn't quite describe that, but maybe you're thinking of the right thing. The reason is, in proline, the nitrogen has 3 bonds to carbon, so it doesn't need any hydrogens. But in the asparagine, the nitrogen only has 2 bonds to carbons, so it needs a bond to hydrogen to get up to 3. Okay, so we got this one incorrect as well. So proline is a little bit tricky. So 2 things to keep in mind about proline are, first of all, the ring includes the nitrogen from the main chain, not a separate nitrogen. The ring includes the main chain. That's the weird thing about proline. And also, unlike the other nitrogens, this nitrogen does not have any hydrogens because it has an extra carbon bond. So we missed those 2 questions. That would be a good thing to mark. There's a good chance you'll be asked to do that on the exam. Proline's an important amino acid. Let's go back to my screen. So let's continue looking at question 6. All right, it looks like you labeled the peptide bonds. Here's the N terminus, here's the C terminus. So what was your answer for 6C? What's the net charge at physiological pH? Now, first of all of your answers were either wrong or at least not reliable, because when you drew your picture, you were drawing the proline incorrectly. In your pictures, the proline was drawn incorrectly. Can proline take 2 different forms, charged and uncharged?
No.
No. So that should have messed up all your answers. That should have messed up all your answers there. So so it looks like we should do that part again, actually. So let's try that again. So you tell me, what's the net charge of the peptide at physiological pH?
Neutral?
That's right. Actually, that's what you said here. I'm not quite sure why you said neutral. You must have, for some reason, left the proline out. So you made 2 mistakes that canceled out somehow there. All right. So in any case, the important thing is to have a reliable method for that. So that's one reason why we have to get the proline correct, so we can have a reliable method for these. By the way, this is a little minor detail here, but notice, do you see what you wrote down here? You wrote 7.4 equals 0.
Mm-hmm.
Do you see how that doesn't make sense?
Yeah.
7.4 doesn't equal 0. So that's something that students often do. They kind of like chain a whole bunch of disparate things up with equal signs when they're not really equal. So I think what you meant to say is when pH equals 7.4, then the net charge equals 0. So who is it that's equal to 0? The pH doesn't equal 0. The net charge equals 0. So that's just it's good to be a little bit more careful with their notation. Instead of saying pH equals 0, we should have said net charge equals 0. Okay. Did you skip the other parts of the problem?
I don't remember why I didn't put an answer there.
Well, let's finish that off now.
Okay.
Your answers might have been incorrect anyway because you were drawing the proline incorrectly. So let's finish this off on your screen now.
equals the sum. I need to use that chart, right?
Pardon me?
That chart from the textbook.
Well, the thing is that you would be expected to have the chart memorized.
Mm-hmm. So it's -2.
Okay, that's good. Those answers are correct. That's good that you saw that cysteine also has two different forms. Now, did you come up with those pKa's from memory, or did you have to check the table?
I need to check the table.
Okay, so obviously that's a job for you to do before you take the exam. I think you did mention that your instructor did say that you needed to have the ballpark figures memorized. This is a pretty sure to be on the exam. It's a very standard type of problem here. One other thing, according to that, since you have that table in front of you anyway, according to that table, what's the ballpark pKa for the N-terminus?
For the N-terminus, you said?
Yes. You can take a look at the table since you have it in front of you.
Oh, so it's not 9, it's 8.
And what's the ballpark pKa for the C terminus, according to the table?
3.
Why did you write down 9 and 2? Well, we talked about this in our last session. 9 and 2 are the pKa's for a free amino acid. But when the amino acid is in a peptide, the pKa's change. So when we were first going over this material, I guess it was a little confusing. When we first went over the material, we did examples about free amino acids. So there we used the numbers 9 and 2. But now we're using, we're doing many problems about peptides. And that's probably what you're going to see on the test. Peptide problems, not amino acid problems. So now we shifted to these pKa's of 8 and 3. I don't know if your instructor would make a big deal about that or not. But in any case, we may as well use these better numbers. So you might want to make a note of that in your notes. 9 and 2 are the pKa's for a free amino acid. But for a peptide, it's better to use these numbers of 8 and 3. That's confusing because we started with 9 and 2, and now we've moved to 8 and 3. Okay, well, I think the other parts, let's finish the other parts here as well. So let's do the rest of part C.
Which means this would be -3.
Who is -3?
The amino acid. The net charge on the amino acid on the polypeptide is -3.
Yeah, the net charge. Okay, that's right. That is correct. Good. Well, let's finish off with the last part, part C.
So +2.
Okay, good. That is correct. Good. All right, let's go back to my screen. Okay. All right, so remember, if you're able to do all those homework problems on your own, then we'll have more time in the tutoring session for new material. So it's always good to try to do all the parts of the homework on your own. That way we don't have to spend the tutoring time on that. But in any case, it's good that you got those questions correct. Number 7. So you said they're rigid because they are planar. Okay, it's on the right track, but we need to clarify that a little. But first, actually, let's see. All right, so do you have that table of ballpark pKa's in front of you right now? Okay, well, I won't spend the tutoring time on this, but that it's important to memorize that table. Remember that you should memorize that table to the closest integer. And arginine is 12.5. No time like the present, so that would be a good thing to do now. Okay, now Let's see. What amino acid is this?
That is serine.
Let's name all the amino acids here. So what's the next one?
That is alanine.
And this one?
That is remember this backwards. Oh, that's glutamine. Hold on, wait. It's not glutamine. That is this asparagine.
What's the difference between asparagine and glutamine?
There's only one CH2 in the or there is no CH2 group in the there's only one CH2 group in the side chain.
For which one?
For asparagine.
And for glutamine?
There's two.
Okay, so which one is this?
That is asparagine.
Okay, that's good that you took your time and came up with a good description of the difference. You came up with a good description, which is asparagine has only one CH2 group in the side chain. What's the three-letter abbreviation for asparagine?
ASP, or ASN.
Okay. Okay, so that's something to keep working on. You need to keep working on having all those amino acids memorized. You should be able to write down the amino acids if you're given the name, but you also need to be able to identify the amino acid if you're given the picture. Those are very important skills to keep working on. All right, so let's continue our discussion of the peptide bond. So we were looking at the peptide bond here. Let's see. So we were discussing what are the characteristics of a peptide bond. So first of all, you said one characteristic is that the peptide bond is rigid. That is correct. That means, is there free rotation around the peptide bond?
No.
No. Why not?
Because it's rigid and double bonds prevent rotation for rotation.
But it looks like it's a single bond, not a double bond.
It's rigidity because it's planar.
But a single bond wouldn't be planar. So it looks like you might have forgotten a little bit the explanation we gave for why peptide bonds are rigid. The idea you're not using here yet is what we talked about last time. There's another resonance structure for the peptide bond. Remember that we drew this other resonance structure. Now in this resonance structure, this is a double bond. So why is it rigid? Because double bonds are rigid and has double bond character because of resonance. So the peptide bond is rigid because there's a resonance structure where it's a double bond. The peptide bond is rigid because there's a resonance structure where it's a double bond. That's why we don't have free rotation, because you don't have free rotation around double bonds. So one key aspect of the peptide bond is that it's rigid. Another key aspect of the peptide bond is that it's planar. The peptide bond is also planar. Why is it planar?
Hold on, sorry.
Sure.
Another reason is because it's
Another characteristic of the peptide bond is that it's planar. We've discussed that it's rigid. Why is the peptide bond rigid?
because it has resonance where there should be a double bond.
Yeah, there's a resonance structure where it's a double bond, and double bonds are rigid. Now, peptide bonds are also planar. What does the word planar mean?
Flat.
And why is it planar?
Because they peptide bonds are planar because they have no sp3 configurations.
I guess that's on the right track. Now they do have an it's sp3 here. In this picture, it would be sp3. You're on the right track. The key is, again, it's because of the resonance. The resonance explains the key characteristics of the peptide bond. It's planar because double bonds are planar. We learned that for alkenes. We know alkenes are planar, and this double bond would be planar too. Both sides of the double bond are planar. So the reason that it's planar is the same reason that it's rigid, because there's a resonance structure where it's a double bond. Why is the peptide bond rigid? Because it has a resonance structure where it's a double bond, and double bonds are rigid. Why is the peptide bond planar? Because it has a resonance structure where it's a double bond, and double bonds in general are planar. So both those things are explained by the resonance. It's planar and rigid, both of them, because of the double bond character in the resonance. Okay. Tell me when you're ready to continue.
You repeat that one more time. I'm going to make sure I have everything.
Okay.
Rigidity, resonance.
Now, the key thing is not just to have it in your notes, but to understand what we're saying. The point here is for it to make sense. So you should kind of be able to check for yourself whether you have what you have down makes sense. But the idea is that peptide bonds are rigid because they have a resonance structure that has a double bond, and double bonds are rigid. And peptide bonds are planar because there's a resonance structure where the peptide bond has a double bond, and double bonds are planar. So that's good to have in your notes, but if you're understanding those ideas, they should make good sense.
Okay.
Okay. What type of functional group do we have here at the N-terminus?
The N-terminus, one second, I can't see.
What type of functional group is it at the N-terminus?
An amine?
That's
or
right.
an amide?
Which one?
Amide?
So this is an amide?
Yes.
And what type of functional group do we have in the peptide bond?
Amine?
All right, we're getting those confused. That's incorrect. That's important to get straight. So let's go back and fix that. A free nitrogen. So first of all, what type of functional group is this?
Is that an amine?
This is an amine. What type of functional group is this?
Amide.
Okay, we were getting those confused a moment ago. Those are important terms to be clear about. So this is an amine and this is an amide. Okay? Are amines basic?
No.
So to
Actually, yeah. Hold on a second. No, they're not.
So do amines have two different forms or only one form?
One form.
So do they have a pKa?
Yes.
Now, anything that has a pKa has to have two different forms. So I think we're getting confused. We must be getting confused. Anything with a pKa has both a protonated and a deprotonated form. So those different answers that we were giving don't make sense. They're not agreeing with each other. Well, actually, amines are basic. Amines are basic. That's very important. What's a base?
Something that accepts hydrogens and donates, or accepts protons and donates electrons.
Okay. So are amines basic?
Yes.
Yes. They're basic because they can donate their lone pair. So what does the other form of this amine look like?
NH3 plus.
That's right. And you would use the ballpark pKa to figure out which form it's going to be in. Now, are amides basic? Well, it looks like we're forgetting some of this, so I'll just answer. Amides are not basic. Amides are not basic. So we need to explain why amides are not basic. If amines are basic and amides look so similar, why are amides not basic? After all, they have a lone pair too. I think we were discussing that in our last session, but let's discuss it again. The reason, again, comes from the resonance structure. If we draw the other resonance structure, remember that when we were doing organic chemistry, we learned how to use the electron pushing arrows to figure out the charges. What should be the charge on this oxygen? What should be the charge on this oxygen in this resonance structure?
Negative.
And what should be the charge on the nitrogen in this resonance structure?
Positive.
Because it started neutral and it's losing electrons. Now, does this positive charge make the nitrogen more or less basic?
Less basic.
Because bases donate electrons. Something with a positive charge wouldn't want to donate electrons. So in fact, the amide is not basic at all. The amide is not basic, unlike the amine, because there's a resonance structure where it doesn't have a lone pair, is one way to put it. The amine is basic because of the lone pair, while there's a resonance structure where the amide doesn't have a lone pair. That's one way to see why it's not basic. Or you can also see from this resonance structure it's not basic because it has a positive charge. If it gained a proton, this resonance structure, well, all right. So, in any case, amides are not basic, and again, that's explained from the resonance structure. The resonance structure explains why the amide is not basic. Remember, the most important factor in organic chemistry is the charges. This positive charge shows how the amide is not basic. So, do amides, this is kind of weird, this is a little hard to phrase, but do amides have a protonated and a deprotonated form?
Apparently no.
No. They have two different resonance structures, but they don't have two different forms the way an amine does. They don't have a protonated and a deprotonated form, because we just said it's not basic. Only acids and bases have two different forms. So does the amide have a pKa?
No.
Yeah, it's only the acidic or basic groups that have pKa's. Okay.
So the amide group itself cannot be positive. It only has a resonance where there is a positive and a negative form of it.
That's true. But even this, what's the net charge on the amide?
Neutral.
That's right. So even it does have a resonance structure where there's a positive charge, but that's balanced by the negative charge. The key point is, notice, what's the net charge on this amine?
Neutral.
And what's the net charge on this amine?
Positive.
So notice the amine has two different forms with two different charges, but the amide only has one form with one net charge. It has two different resonance structures, but that's not a new charge. Now, that's something we've been taking for granted all along, because, for example, on the problems that you were just doing, for example, here, notice that when you were writing out this whole structure, you said this nitrogen is protonated. But is this nitrogen protonated?
No.
Why not? Because it's an amide nitrogen.
Mm-hmm.
Amine nitrogens protonate, but amides don't. Is this nitrogen protonated?
No.
Because, again, it's an amide. So we've been taking for granted all along. We've been taking for granted all along that the nitrogens in the peptide bonds only have one form. It's only the N-terminus that has two different forms. And now we've simply explained that. Okay, so now we can summarize the key facts about peptide bonds. So one key fact about peptide bonds is that they're rigid. Why are peptide bonds rigid?
Peptide bonds are rigid because they are they have resonance where there should be double bonds, and because they're planar because of resonance.
Okay, that's right. Those are actually two different things. They're rigid because of the double bond resonance structure. And why are they planar? Again, because of the double bond resonance structure. And are amides basic? Are amides basic?
No, they're
Why
not
not?
basic
Why not?
because they have a resonance. They have a resonance of two different structures of net charge of neutral.
Why does that mean they're not basic the way an amine is basic?
Because the amide cannot have a positive charge.
Let's see, we haven't quite put our finger on it there. So the point here is amines are basic. There's two ways to look at it. Amines are basic because they have a lone pair. But amides are not basic because one of their resonance structures does not have a lone pair. Or another way of looking at it is amides are not basic because they have a resonance structure where the nitrogen has a positive charge. Well, once you have a positive charge, you don't want to gain another proton. I hope that's making sense. Remember, we're not trying to just memorize this blindly. We're trying to understand the ideas. So I hope all of those ideas are making sense. So all of the key ideas about the peptide bond make good sense. What I'm trying to point out here is all of the key characteristics of the peptide bond make good sense based on this resonance structure. The peptide bond is rigid because of the double bond in this resonance structure. The peptide bond is planar because of the double bond in this resonance structure. And the peptide bond and the nitrogen in the peptide bond is not basic because it has a positive charge in this resonance structure. Okay, I hope that's making sense. All right, so those would all be good things to have in your notes. Those are important points about peptide bonds. So let's continue. Let's see. What's the name of this amino acid?
That is an alcohol.
It is an alcohol functional group, that's right. But what's the specific name of this amino acid?
That is it's not serine. It's not serine. That is. Oh, that's threonine.
That is correct. Okay, that is correct.
Serine the whole time.
What's the name of this amino acid?
Leucine.
That's right. By the way, another name for an amino acid in a peptide is a residue. How many residues are in this peptide?
3.
Good. So that's just a term we haven't used before. Residue is just an amino acid part of a peptide. What's the name of this residue?
That residue is glutamine.
What's the three-letter symbol?
GLU.
Like this?
GLN.
Like this. That's right. Okay. All right. All right. So it looks like you could still use some work on the amino acids. You had a little trouble with some of these. Remember, the best thing is just write out the whole list once a day. Write out all 20 of them once a day so that you're very comfortable with those. Keep in mind, your course has gotten far beyond where we are right now. Your course is far beyond the chapter we're on here. So we need to be able to move more quickly through this. So that means you need to put in the time on your own to have this material memorized. So we don't need to spend time on it in the tutoring, and we can kind of catch up with your course, hopefully. Okay, so let's see. Is this the peptide bond?
No.
Is this the peptide bond?
Yes.
Yes, this is the peptide bond. Is there free rotation around this bond?
No.
Why not?
Because there's resonance there, and it's planar.
Now, it still sounds like you're kind of just repeating the words without thinking about what they mean. It's not rigid because it's planar. What is it about the resonance structure that makes it rigid?
It has a resonance where there should be a double bond.
Yeah, it's the double bond that makes it rigid. That's important. Remember, this is a thinking class. We don't want to just be blindly memorizing the chains of words. We want to understand why the concepts make sense. So why is this rigid?
Because there's resonance where there's a double bond.
And double bonds don't have free rotation. Is this a peptide bond?
Yes, yes.
Is there free rotation around this bond?
No, because there's double bond. There's resonance where a double bond should be.
And double bonds don't have free rotation. Okay. Is there free rotation around this bond?
Yes, yes.
Why?
Because there is no resonance where a double bond should be, and thus after rotation.
So is this a single, double, or triple bond?
It's a single bond.
And there's free rotation around single bonds. Okay. So, is this part of the peptide going to be rigid or flexible?
That is going to be rigid.
And will this part be rigid or flexible?
Flexible.
So, in the peptide can rotate around this bond. Since we can rotate around this bond, the substituents on the bond can take different angles. So we need a name for this angle. The angle of rotation around this bond is called for some reason I'm blanking out and forgetting how to draw. All right, there it is. Actually, it does look a little bit like the symbol for 0. I'm trying to draw it so it doesn't look quite like a 0. Okay, flanking out on how to draw this. But anyway, it's used this symbol from the Greek alphabet, which is phi. So remember, the peptide can rotate around this bond. So the substituents on around this bond can take different angles. The angle of rotation around this bond is called phi. Okay, so that's something you should probably know. That was in one of the handouts that you sent me that your instructor had talked about. Okay, so this is called phi. The Greek letter phi. Is there free rotation around this bond?
Yes.
Why?
Because single bonds have free rotation and there's no resonance there.
So there's no double bond character. Okay, so there must be a name for the angle of rotation around that bond. I don't know if you happen to remember from class what this is called. So the symbol here is psi. This is phi, and this is the Greek letter psi. Tell me when you have that down.
Yep.
Is there free rotation around this bond?
No.
Is there free rotation around this bond?
Yes.
What's the name of the angle of rotation?
That is phi.
Good. It could be pronounced phi or phi. Good. Is there free rotation around this bond?
Around.
Sorry. This one?
Yes.
What's the name of the angle around this bond?
Psi.
Is there free rotation around this bond?
No.
How about this one?
Phi.
How about this one?
Psi.
How about this one?
No.
How about this one?
Phi.
How about this one?
Psi.
How about here?
No.
Okay. So I think you might be tested on these names. So do you remember in the past we talked about how the slogan to memorize is nitrogen, alpha-carbon, carboxycarbon, nitrogen, alpha-carbon, carboxycarbon, nitrogen, alpha-carbon, carboxycarbon, nitrogen, alpha-carbon, carboxycarbon. Well, now we can do the same thing for the bonds. So notice that the bonds here, you can see you have peptide bond, phi bond, psi bond, 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 peptide bond. So that might help you to memorize those names and symbols. So those should be memorized. Okay. Do you have that down?
Yeah.
All right. There's a lot of memorization in this class, so when I mention things that have to be memorized in the tutoring, that's something to make a note of and make sure you do that, because by the time of the next session, we'll have more stuff to memorize. All right. Now, does this phi have to be the same value as this phi? And the answer is no. There's no reason why this bond has to be rotated the same amount as this one. All of these bonds could be rotated different amounts. So all of these phis could be different numbers, and all of these psis could be different numbers. So that's one of the reasons why proteins are so can take on such a wide variety of shapes, because they have a wide variety of rotations they can take on around these bonds, which allows them to accomplish many functions in the body. Can this bond take on any possible value? So theoretically, the possible values for phi go from negative 180 degrees to positive 180 degrees. If you go from negative 180 to positive 180, that covers the entire range of possible angles. But are those all really possible? Can this take on all the possible angles? And the answer is no. Because of steric hindrance from the side chains, only certain values are possible. Some forms of rotation would push the two side chains too close to each other. You can kind of see that right now there's a good, the rotation is very, we have good levels of rotation with the side chains far separated from each other, at least on the page here. But if you rotate or fold the molecules in a certain way, there would be too much steric hindrance. So only certain angles are allowed. The angles that are allowed for the bonds are called the Ramachandran plot.
What?
The allowed angles from the plots, the allowed angles for the bonds are given in a Ramachandran plot. Now, this is a pretty typical topic in biochemistry, but from your reaction, it's sounding like your instructor didn't talk about this. Do you remember this being talked about in class?
No, not at all.
Okay. All right. This is a very standard topic, but if your instructor didn't talk about it, then we don't need to, hopefully it won't be on the test. Okay, so I won't go into that. Your instructor, I did talk about phi and psi. I saw that in some of the handouts he sent me. But I didn't see anything specifically about the Ramachandran plots. But I do think they want you to know that the names of these angles are phi and psi, and you should keep in mind that only certain angles are allowed, and the angles that are allowed can vary depending on the side chains. So let me ask you this. Which amino acid do you think would have the most allowed angles? Which amino acid would have the most allowed angles?
The amino acid with the most allowed angles would be glycine, maybe?
That is the correct answer. Yes. Why is that?
Because there's less steric hindrance and there's, I guess, more room for rotation.
Yeah, that's right. Remember that one of the main things that prevents amino acids from rotating to certain angles is that their side chains would bump into the neighboring side chains. But glycine doesn't have to worry about that too much because its side chain is so small. So glycine has the most allowed angles of all the amino acids. What amino acid do you think might have the fewest allowed angles?
The fewest allowed angles would be probably tyrosine or tryptophan.
Those are good guesses. Those are good guesses because they have so much steric hindrance. That's right. I think, though, that the one that's the most rigid is proline. Remember, we've been talking about how proline introduces rigidity. So your answers were good ones, too. Those probably have a lot of restrictions. But I think proline is the most restricted because, remember, it has that ring that's part of both the side chain and the main chain. Remember, we talked about that in the homework, how proline introduces extra rigidity. So you're probably right that the amino acids with great big rings in the side chain have fewer allowed angles. But I think the one with probably the fewest allowed angles is probably proline. Okay. so moving on. What are the 4 types of amino acid side chain?
4 types of amino acid side chain.
Remember that your textbook groups the amino acids into 4 groups. What are the 4 groups?
Oh, positive charge, negative charge.
Positive charge. Polar and nonpolar.
Okay. So those are, remember, I'm hoping that you're learning the amino acids in those groups. You should know which group each amino acid falls into. I guess to save time, I won't quiz you on that, but that's important to keep in mind. Okay, so that was actually review. Now moving forwards, there are 4 levels of protein structure. There's 4 levels at which we can describe the structure of a protein. And the names of those 4 levels are here's the first 2 names. What do you think is the next name?
Tertiary, quaternary.
So that the chemists didn't exactly outdo themselves in the creativity department here when they were naming these. But these are certainly logical names because they're increasing, in a sense, increasing levels of complexity. Okay, have you learned about these in your course?
Yes.
Let's see. So let's, for example, let's look at this peptide here. What's the primary structure of this peptide? What is the primary structure of this peptide?
Primary structure is polypeptide, or
so if you let's say that was a question on the test. If the test asked you what is the primary structure of this peptide, what would you write down as the answer?
SER-ALA-ASN.
All right, that's what I was going for. So notice primary structure is a very simple idea. The primary structure is just the sequence of amino acids. The primary structure is just the sequence of amino acids. So the primary structure is this. Very good. This is the primary structure. Excellent. Could you write it like this? Is this the primary structure?
No.
How do you know which way to write it?
From the N-terminus to the C-terminus.
It's always got to be from the N-terminus to the C-terminus, regardless of how the picture is drawn. This picture happens to have serine first, but that doesn't mean, that's not the reason that serine is first over here. The reason that serine is first here is because that's the N-terminus. Usually the pictures are drawn with the N-terminus on the left, but if the N-terminus had been on the right, you would still start with the N-terminus when you're describing the primary structure. Primary structure is always described from N-terminus to C-terminus. That's absolutely crucial. It seems like you might have that down. All right, so what's the primary structure of this amino acid?
Just that would be threonine dash leucine dash glutamine, or glutamine dash glutamate dash histidine.
What comes after the glutamine?
The histidine.
Take your time. What comes after the glutamine?
Oh, glutamine, you said?
Yes.
Glutamine.
What comes after the glutamine?
Oh, I didn't see that one.
cysteine, and then the glutamate, and then the histidine.
Okay, so at first you left out this one. Remember that usually when the molecules are drawn this way, the side chains alternate above and below. What's the name of this one again?
That is cysteine.
You have to be a little bit more careful about that. It's pronounced cis.
Cysteine.
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 Cysteine. That is, you have to pronounce the E separately from the ine. And the only reason I'm making a big deal about that is that there's another name. There's something else in biochemistry that is pronounced cysteine. Cysteine is actually something else we'll talk about pretty soon. So cysteine is not the same as cysteine. So we actually have to be pretty careful to pronounce this E, which is pronounced cysteine here, because it's actually not the same as cysteine. Cysteine is a little bit more complicated thing that we'll talk about later. So what's the name of this residue?
That residue is cysteine.
Cysteine, good. Okay. All right, very good. So this is a primary structure. There's a good chance you'll see a question like this on the exam. You might be given a problem where the structure is drawn for you, and you might just be asked to write down the primary structure. Well, primary structure is a very simple, very straightforward idea. It's just a listing of the amino acids. The only tricky thing is they have to be listed in a certain order. Which terminus comes first?
The N-terminus.
Yeah. When you draw an amino acid, you don't have to put the N-terminus on the left. But when you write down the primary structure in letters, then you have to put the N-terminus on the left. That's very important. Okay, well, there's not too much more to say about that. Let's see. Okay. All right. Well, that's all I had to say about primary structure. So let's go on to secondary structure. So there's two main types of secondary structure. This is a higher level of structure. There's two main types of secondary structure. What are the two main types of secondary structure?
The two main types are alpha helix and beta-pleated sheet.
Excellent. That's right. Also, another type of secondary structure are certain types of turns or loops. I didn't see your instructor going into too much detail about that, though, so I'm going to mainly focus on these first two. So various types of turns and loops are considered secondary structure as well. So a secondary structure is a regular repeating structure. Secondary structures have repeating patterns, regular repeating patterns. Alpha helix and beta-pleated sheets have regular repeated patterns. So now we should talk about that. Let's see. So let's start with the alpha helix. So this would be part of the larger protein. Part of the protein could curl into a helix. So it'll simply look like a helix. Let's see. Notice here's the angles we were just talking about. What's the name of this angle?
Phi.
And this one?
Psi.
Okay, so it looks like your instructor must have talked about that somewhat, but maybe they didn't get into the Ramachandran plots, but they did label what these angles were. All right? Now here's what we're talking about now. What's the name of this secondary structure?
Alpha helix.
That's right. By the way, do you know what the name of this secondary structure is?
That's also alpha helix.
It's harder to see, but if you could see the perspective here, this is also in a helix. Do you know what this one is?
That's a beta-pleated sheet.
Does it look like a sheet?
Oh, is that the view of the alpha helix from above?
Yeah, this is the alpha helix if you were kind of like looking down from above. So this is still the alpha helix from above. notice that, okay, this is also the alpha helix from above. Any idea which one this was? D.
Is that the orbitals of it?
So this is also the alpha helix. This is a space-filling model. This is just to show that this is kind of unrealistic. In reality, the interior of the helix is filled with atoms, is the point they're making. There isn't really space in the middle of the helix like you might think here. It's all filled with the atomic radiuses of the atoms. Okay, so your instructor gave you all these pictures, but I think this is the one that's easiest to understand. So let's focus on A. This gives us the basic picture here of a helix in A. Here's our helix. Now, things don't naturally take helical forms unless there's something to hold them this way. So this is something you've studied in class. Do you have any idea what's the main thing that holds the helix in this shape? What holds the helix in this shape?
The polypeptide bonds?
Could you repeat that?
Is it the polypeptide bonds?
Let's see, I'm not quite sure what you mean by polypeptide bonds. That's not really a term that we've used before. Were you thinking just of the peptide bonds? Now, that can't be right because peptide bonds just hold together the amino acids in the primary structure. Peptide bonds just hold together the primary structure. They can't explain why this would take the form of a helix, why it would take this form. Maybe we should kind of review some of the basic types of bonding here now. This is important in the course. One key type of bonding is covalent bonds. This is from an earlier chapter. We didn't kind of go into this in detail. So there's covalent bonds. Then another type of bonding are weak interactions. Who do you think is stronger, covalent bonds or weak interactions?
Covalent bonds.
You can kind of tell from the name that weak interactions are kind of weak. There's 3 key types of weak interactions that your textbook covers. Do you remember any of these? What's a type of weak interaction?
Hydrogen bonds, van der Waals, and London. I forgot the name of it.
Actually, your textbook doesn't use the term London forces. Maybe your instructor used that. I think London forces are pretty much the same thing as van der Waals forces. London forces are pretty much identical to van der Waals forces, so I'm not going to write them down separately. London forces are pretty much the same thing as van der Waals forces, so I'll just stick with the one term, van der Waals forces. So the third type here we haven't quite named yet. The third type is electrostatic interactions. So these are very important concepts in your course. Any idea what we mean by an electrostatic interaction? This would be between what 2 things?
Between something that's negatively charged, something that's positively charged.
Excellent. That's exactly right. Yes. So an electrostatic interaction is a attraction between an atom with a positive formal charge and an atom with a negative formal charge. Tell me when you have that down.
Okay.
So can you think of 2 side chains? What are 2 side chains that could form an electrostatic interaction?
Positively charged side chain and negatively charged side chain.
Excellent. That's a good start. So can you give me a specific example of 2 side chains that could form an electrostatic interaction?
You can have glutamate and arginine.
Okay, that was a good example. Arginine and glutamate. And we could draw the electrostatic, I didn't leave myself too much room here, but here's the electrostatic interaction between those. Notice that for weak interactions we use dots or dashes. So we use these dots to show the electrostatic interaction between the positive charge and the negative charge. Can you see what I'm trying to show with this notation? I'm trying to say that this is a peptide strand, but I don't want to take the time to write all the amino acids in it. So this squiggle just means that the peptide strand continues over here, but I was too lazy to draw what's over here, and I was too lazy to draw what's over here. And here's another peptide strand, but I only drew one part of the peptide strand because it would be too time-consuming to write the whole thing. Okay, so here's an electrostatic interaction. All right, very good. What type of interaction is this? What would be the name for this? That's covalent bond. That's right. So this right here, this is a covalent bond. How about this?
Covalent bond.
How about this?
Covalent bond.
How about this?
Covalent bond.
In fact, all the solid lines are covalent bonds. Solid lines are always used for covalent bonds. Dotted lines are used for the weak interactions. So who's stronger, a covalent bond or an electrostatic interaction?
Covalent bond.
Yeah. At least in an aqueous solution like the body, the covalent bond will be stronger. Okay. Give me another example of two amino acids that could form an electrostatic interaction.
Aspartate and lysine.
Excellent. Good. So that's one reason why we need to know who's charged, so we can figure out who can form these types of electrostatic interactions. Are there any other negatively charged side chains?
Negatively charged side chains? I don't think so, no.
Are there any other positively charged side chains?
Histidine?
Yeah. Now that's borderline because remember histidine is only 10% charged, but at least it has the potential to have electrostatic interactions 10% of the time. Histidine would have a 10% of the time it could form an electrostatic interaction with a negative charge. So that might be enough. That might be enough to be a stabilizing force for a part of a peptide. Or you might alter the conditions so the histidine is more protonated than usual, so you can form the electrostatic interaction. Okay, so that's an important idea about electrostatic interactions. How about hydrogen bonds? What's a hydrogen bond? When can you, between what things do you form a hydrogen bond?
When water is made of hydrogen bonds.
What do you mean when you say water is made of hydrogen bonds?
The oxygen is bonded with hydrogen through hydrogen bonds. Is it anything that hydrogen bonds to is hydrogen bond?
So what type of bond is this?
Hydrogen bond.
Okay, no, that turns out to be a mistake. So that's important for us to take our time and clarify. That's a very important idea. So we need to, so it looks like we haven't really quite learned what hydrogen bonds are. You and I haven't talked about that before, so no time like the present. We need to clarify what a hydrogen bond is. First of all, remember, hydrogen bonds are a weak interaction. They're not covalent bonds. So what type of bond is this in water?
No, it's covalent.
Yeah. Do you remember we were just saying that all the solid lines indicate covalent bonds? In normal chemical structures, solid lines are always covalent bonds. All the solid lines are always covalent bonds. So what type of bond is this?
Covalent.
Is it a hydrogen bond?
No.
No. Because it's a covalent bond. Okay.
Oh, I know what it is now.
Okay. Try again. Go ahead.
It has to do with adhesion and cohesion, so it's is that related to adhesion and cohesion?
Okay, it is related to adhesion and cohesion. That's an application that won't be very important for us in biochemistry, however, although they are related. So let me just help you with that. A hydrogen bond is a weak attraction between a delta positive hydrogen and an atom with a delta negative or formal negative charge. You can tell me when you have that written down.
Okay.
I put this part in parentheses because it doesn't seem to come up very much. Usually this is an attraction between a delta positive and a delta negative. I think it would still be considered a hydrogen bond if it was a full negative, but I put that in parentheses because it doesn't come up very much. Most of the time we'll be focusing on delta positive hydrogen and another atom with a delta negative charge. What is the delta charge on this hydrogen? Positive, delta negative, or neutral?
Neutral.
What's the delta charge on this carbon?
Neutral.
How do you know?
Because it has 4 bonds, and it's not electronegative.
Let's see. I don't think we've quite put our finger on it. No, because carbons could have delta charges, so we haven't quite put our finger on it. Here's the key. Is there an electronegativity difference between carbon and hydrogen, or do they have similar electronegativity?
They have similar electronegativity.
How do you know? Does the periodic table tell you that? The periodic table doesn't tell you that, but I think we just memorize that hydrogen is similar to carbon in electronegativity. So who's going to pull, are the electrons going to be pulled closer to the carbon or closer to the hydrogen, or will they share equally?
Equally.
Well, if they're going to share equally, I'll put the 2 electrons right here in the middle. That means neither of them is going to be charged. What's the delta charge on this hydrogen?
Delta positive.
And on the oxygen?
Delta negative.
How do you know?
Because oxygen is highly electronegative, so it's pulling on the hydrogen's electrons more than the hydrogen is pulling.
So the electrons in the bond would really be, say, over here, closer to the oxygen, which would give it the negative charge. How do you know that oxygen is more electronegative than hydrogen? Well, who is hydrogen similar in electronegativity to?
Carbon.
And oxygen is to the right of carbon in the periodic table. Okay, now let's say I took another of this same type of molecule. What's the delta charge on this hydrogen?
Delta positive.
And here?
Delta negative.
So this oxygen would be attracted to which part of the other molecule? This oxygen is attracted to which atom in the top molecule?
The hydrogen.
Because unlike charges attract. So there would be an attraction here. Do you know what the name of this attraction is?
Van der Waals.
Actually, this is a hydrogen bond. This is a hydrogen
okay.
bond. Does that make sense? Remember, what did we say a hydrogen bond was? We said a hydrogen bond is an attraction between a delta positive hydrogen and an atom with a delta negative charge. Well, notice that's exactly what we have here. An attraction between a delta positive hydrogen and an atom with a delta negative charge. So this is the hydrogen bond. Here's another molecule of the same type. So what would this oxygen be attracted to in the neighboring molecule?
Which that would be attracted to the hydrogen in the molecule of the hydrogen bond.
Okay. What type of attraction is this?
Covalent.
How about this?
Covalent.
How about this?
Covalent.
How about this?
Covalent.
Is this a hydrogen bond?
No.
So if you're bonded to a hydrogen, does that make it a hydrogen bond?
No.
So I don't know if you would agree, but it seems like the chemists did not do a great naming job here. This certainly looks like a bond to hydrogen, but is it called a hydrogen bond?
No.
Doesn't seem very logical. So this is not a hydrogen bond. That's not what this... so a hydrogen bond is not a type of covalent bond. This is not a hydrogen bond. What type of bond is this?
Covalent.
Right. I'm emphasizing that because I think we were getting confused about that earlier. What type of bond is this?
Hydrogen.
How about this?
Covalent.
How about this?
Hydrogen.
How about this?
Covalent.
Is this a hydrogen bond?
No.
No, okay, good. So that's very important. Who's stronger, the covalent bond or the hydrogen bond?
Covalent.
Okay. All right, so what is a hydrogen bond?
Hydrogen bond is weak interactions or attractions between a delta positive hydrogen and an atom with a delta negative or formal negative charge.
That's right. Usually, almost all the examples you see, I think, will just have a delta negative. But I suppose it would work if it was a full negative. It would be even stronger then. Okay, good. What's the name of this molecule?
Water.
Yes. Can this water form a hydrogen bond with this water?
Yes.
Is this the hydrogen bond?
No.
Is this the hydrogen bond?
Yes.
Yeah. For that matter, we might form another hydrogen bond. There can be tons of hydrogen bonds. Water has special properties because it has the potential to form lots and lots of hydrogen bonds. You were mentioning adhesion and cohesion. Those are based, I think, on hydrogen bonding, but I don't think we need to get into those details because that's not an application you would focus on in your biochemistry course. Okay. A few more terms. So in this picture, this molecule here is considered the hydrogen bond donor, and this would be the hydrogen bond acceptor. So the molecule that has the hydrogen is the hydrogen bond donor, and the molecule without the hydrogen is the hydrogen bond acceptor. So for this bond here, is this molecule the hydrogen bond donor or acceptor?
The donor.
How about this one?
The acceptor.
How about for this bond? For this bond, Is this molecule the hydrogen bond donor or acceptor?
Donor.
How about this one?
Acceptor.
For this hydrogen bond, is the top water the hydrogen bond donor or acceptor?
Acceptor.
And the bottom water?
Donor.
Because it's donating the hydrogen. How about for this bond? Is the hydrogen on the left the hydrogen bond donor or acceptor?
The donor.
And on the right?
Acceptor.
What's the main fluid? What's the main molecule in the fluid in the body?
Water.
Yes. Can you think of any side chains that can hydrogen bond with water? What would be a side chain that could hydrogen bond with water?
Polar molecule. Polar one.
Excellent. So give me an example.
Tyrosine. Actually, is yeah.
Is this the hydrogen bond?
No.
So this, for example, would be the hydrogen bond. Okay, good. Does that tend to make tyrosine relatively hydrophobic or hydrophilic?
That makes tyrosine hydrophilic.
Because it can form better interactions with the water. Good. Can you give me another example of a side chain that could form hydrogen bonds with the water?
Serine.
Is this the hydrogen bond?
No.
Okay, that's a good example. Give me another example of a side chain that can form a hydrogen bond with water. Actually, let's go back to serine for a second. So is serine hydrophobic or hydrophilic?
Hydrophilic.
Especially because of these hydrogen bonds it can form. Okay, can you think of another example that could form hydrogen bonds with water?
Cysteine.
Okay, that one's a little bit borderline because who's more electronegative, oxygen or sulfur? Hello?
Hello.
Hi.
Yeah.
All right, well, fortunately we haven't been getting cut off too much today, so one time's not so bad. Let me turn on my screen again. I was asking who's more electronegative, oxygen or sulfur?
Oxygen is more electronegative.
Because do you remember the things that are most electronegative are the things that are furthest to the right and high in the periodic table. So who has the bigger delta negative, oxygen in this bond or sulfur in this bond?
Oxygen.
That's right. So the delta negative here is a smaller delta negative. And who has a bigger delta positive, the OH hydrogen or the SH hydrogen?
SH.
Because, excuse me?
The OH hydrogen.
Is that bigger or smaller delta positive?
The OH has a bigger delta positive.
Because this oxygen is pulling the electrons to it more than the sulfur is. So I would say the hydrogen bonding here is probably less effective, but I think in your biochem course you might still say there's a hydrogen bond there. Okay, so maybe a slightly less hydrogen bond, but there's still some hydrogen bonding potential there. Okay. What's the name of this side chain?
methionine.
Yeah, maybe this isn't the best example. That's a little bit borderline. Let's do something that's not borderline. What's the name of this side chain?
Valine.
Can it form hydrogen bonds?
No.
Okay. Some of them are a little bit borderline, but clearly this side chain here cannot form hydrogen bonds. Why can this not form hydrogen bonds?
Because it is nonpolar.
Okay.
And it doesn't have an atom with a delta negative charge that can form hydrogen bonds.
Or a delta positive hydrogen. Either of those could form a hydrogen bond. It also doesn't have any delta positive hydrogens. Why does it not have any delta positive hydrogens?
Because the carbon and hydrogen have the same almost or similar electronegativity.
Okay, so it's important to be able to recognize which side chains. So would this tend to be hydrophobic or hydrophilic?
Hydrophobic.
Because it can't form good interactions with the water. Okay, good. So what is a hydrogen bond?
A hydrogen bond is an attraction between it's a weak attraction between a delta positive hydrogen and an atom with a delta negative or formal negative charge.
Okay, very good. And again, I think mainly you'll see examples with delta negative charges. I just included the formal negative charge for completeness, but that's good. So are the bonds inside of a water molecule hydrogen bonds?
No.
What role do hydrogen bonds play in water?
Hydrogen bonds are what keeps water fluid.
Let's see. Can you be? It's not quite right. Can you be a little more specific? Where are the hydrogen bonds in water?
Hydrogen bonds are between the hydrogen and the oxygen of different water molecules.
That is exactly what I was going for. Yes. So I think that's something we weren't understanding at the beginning. When I first asked you about hydrogen bonds, it sounded like you thought that the hydrogen bonds were inside the molecule. It sounded at first like you thought like the bonds inside the molecule were hydrogen bonds. But now I think we have that correct. This is not a hydrogen bond. The hydrogen bond in water are the bonds between separate molecules. Hydrogen bonds are not always between separate molecules. Hydrogen bonds could be between separate parts of the same molecule. For example, one part of a peptide could hydrogen bond to another part of a peptide. But in water, the hydrogen bonds are between two separate molecules. Okay, that's a very important concept in biochemistry that I think we were confused about a few minutes ago, so that's good that we were able to clarify that. Hydrogen bonds are not covalent bonds. They're a weak attraction between a delta positive hydrogen and an atom with a delta negative. So this is a very important table that we're going over for the rest of the course. So we're about out of time. But getting ready for your next session, it's very important to memorize what we talked about here. What I'm going to say is you should memorize this table because we're going to need to build on that as the course goes on. These are very important ideas. What we need to do is see how these interactions stabilize secondary and tertiary structure. A very important topic is to understand which of these interactions are involved in secondary and tertiary structure. But we can't do that unless we know what these mean. So we need to make sure we know what these ideas are. All right, so we tried to talk about the peptide bond today. We talked about how the resonance structure makes it rigid, planar, and not basic. We talked about these angles. And now we started getting into the levels of protein structure, but then we had to kind of make a digression. We had to digress from protein structure because protein structure is maintained by different types of bonds and interactions. So we can't discuss the different types of structures unless we understand these types of bonds and interactions. Like I say, make sure that you have this memorized so that we can build on this. It's very important to review carefully what we talked about in today's session to make sure that it's clear in your minds. I hope that you're reviewing the tutoring notes after each session. And also, you should be reviewing the previous tutoring notes because all that earlier stuff is important too. Also, I've mentioned to you there's a bunch of things you need to have memorized, so you need to keep working on that. And as usual, I'll send you a homework assignment. Remember to review the tutoring session carefully. Remember to try to do all the homework problems that I'm going to send you ahead of time. You should keep in mind this is a challenging course and it takes a major investment of your time outside of the tutoring, reviewing the tutoring sessions, doing the homework problems, memorizing stuff.